Thanks to all of you for joining in today on our research showcase. My name is Dr. Heather Ray. I'm actually brand new to ISU and the Department of Biological Sciences, so I'm just now getting my lab set up, but I'm happy to have this chance to share with you a little bit about the type of research that we do in my lab and will do in my lab, and some of the types of questions that we like to ask. So, I am a developmental biologist, okay, which really just means that I am fascinated by the process of embryonic development. And the fact that we start out life as one single cell, this is the fertilized egg that we call a zygote, and that 9 months later, we show up, right? We're born as a living, breathing, functioning, multicellular organism. So how does this even happen, right? There's so much that has to happen. This is an incredibly complex process to go from point A to point B. So if we think just from the perspective of the cells in the body, cells are having to make a lot of decisions during embryonic development. Some of these decisions include deciding what eventual cell type the cell wants to become within the body. We need to create a lot more cells. They're having to always replicate and proliferate, but each cell type needs to kind of determine how many of that particular cell type is needed within the body. Cells need to be able to communicate with each other so that in the end we have tissues and cells that are able to coordinate important processes within the body. And also during development, frequently cells have to leave one site within the embryo and actually move and migrate to another place within the embryo in order to set up shop. So all of these cell decisions are really important for ultimately forming tissues and coordinating within organ systems. But if we kind of dive deeper beyond the cell decisions, we see that all of those decisions are driven by gene expression. So a particular gene X is going to give the instructions to the cell to carry out these different cell decisions. And so you can imagine then if there is a mutation that happens in a particular gene, that is going to disrupt the decisions that that cell is making, and that ultimately disrupting those cell decisions will disrupt tissue formation. And unfortunately, this actually happens quite a bit in the human population, so that mutations within genes in early development will end up leading to babies that are born with different developmental disorders, which can have a huge impact on the health of that individual. So in my lab, we're interested in kind of studying both sides of this same coin and really asking kind of a couple big picture questions then. First is, how do genes instruct cell decisions during development? And then the second, how does mutation in a particular gene disrupt those cell decisions, leading to developmental disorders? So just in the last couple minutes, I wanted to introduce one particular ongoing project in the lab, just to give you an idea, again, of some of the work that is being done in the lab. Oh, I'm sorry, I skipped a whole section. I'm used to having my presenter view. Let's go back. Sorry. Okay, I'm asking these big picture questions, right? And ultimately, I am interested in understanding how these processes happen in human development and how developmental disorders arise in humans. But as you can imagine, we can't really study embryonic development in the human embryo inside of the womb. So we need to have a surrogate system to be able to address these questions. So the surrogate system that I have chosen in my lab is the African clawed frog, Xenopus laevis. Okay, these are two beautiful Xenopus ladies here. And at first glance, you might look at them and think, well, these frogs don't have a whole lot in common to humans, but that is not the case. So we actually share a good bit in common. So first, frogs are vertebrates, which means that we share a lot of the same tissue and organ systems with each other. And secondly, a lot of research over the years has found that both the genes and the cellular processes that are involved in very early embryo development are shared between humans and frogs. Another big advantage for us is that frogs lay eggs, and then the embryos develop in the external environment outside of the mother. So we can induce the frogs to lay eggs and then do in vitro fertilization. So you can see the little embryos here, and they will develop externally out on the bench top, and we can actually watch development happen in real time and have access to these embryos in order to do our experiments. And then finally, if you want to study how genes are involved in a process, you want to be able to manipulate gene expression. And we can do that in our frog embryos because we have access to them. So we use a contraption that looks like this with a stereo microscope and a really fine glass needle, and we can inject substances directly into the embryos. So we can inject mRNA in order to overexpress a specific gene, or we can inject what's called an antisense morpholino, and that will knock down expression of an endogenous gene. And so then we can follow along in development and see what the consequences of that are. And also in Xenopus embryos, we are able to actually, through our injections, target specific tissues in order to only affect gene expression in one part of the body and not others. Okay, so now I'm going to tell you a quick little story that we are currently working on in the lab. And that's related to a gene called MASP1-3. So we've been asking the question, what is the function of MASP1-3 during development, and how does mutation in MASP1-3 lead to 3MC syndrome? So MASP1-3, what is it? It is a serine protease, so this is an enzyme. And interestingly, mutations within this gene were identified in several patients that have a syndrome called 3MC syndrome. This is a rare autosomal recessive, so inherited developmental disorder that comes with a number of different phenotypes. These include changes in the shape of the facial structures, cognitive impairment, hearing loss, and a number of others. But what's interesting is that MASP1-3 is really known only for its function in the innate immune system. And so the question arises then, what is the job of MASP1-3 in development? But because this mutation in this gene has an association with the developmental disorder, this suggests that it actually plays a really important function during development. So we've been studying this gene in our Xenopus system. So these are early Xenopus embryos at different stages of their development. And this is a type of experiment called in situ hybridization, where we can actually look at where a specific gene is actively being expressed at any particular time. So what we found was that MASP1-3 is expressed in the developing brain region, starting at the very earliest stages of brain development and continuing on throughout brain development. Okay, and then we knocked down expression of MASP1-3 in these embryos and found that it does disrupt development, especially in the head tissues. And so together this suggests then that MASP1-3 may play an important role in early brain development. And then finally, we've been starting to look at what the function actually is of MASP1-3, what genes or what proteins it may act on as a protease. One experiment that we did, we found that when we overexpress MASP1-3 in the early embryo, we get this massive increase in expression of another gene called BMP4. And BMP4 is known to be really important for the earliest stages of brain development, and the proper expression levels of BMP4. So again, this goes with our other data that MASP1-3 may be really important for early brain development, and so mutation within this gene could disrupt that brain development, and that could be the cause of the cognitive impairment phenotypes that are seen in the 3MC syndrome patients. Okay, and just want to thank a couple students in particular that did this work that I showed you today that were from my lab in Alabama. Yihan Zhang and Nazia Jamil were great undergraduates that worked with me, and my current lab members here at ISU. Thank you.